Gram stain is used for separating gram-positive bacteria from gram-negative. So if you've noticed, there are some structural differences between gram-positive and gram-negative, as a consequence of which the staining for these two bacteria, these two groups of bacteria, will be different. So in gram-positive bacteria, the purple crystal violets that is present in the gram stain will mark the peptidoglycan wall, and thus these bacteria will have a purple color. While in gram-negative bacteria, since the peptidoglycan wall is so small, it cannot retain the purple crystals and will be later on stained with a safranin, which has a pink color. So these bacteria will present with a pink color versus the purple color, which was presenting due to the crystal violet stain of the peptidoglycan. So in any case, one main difference between gram-positive and gram-negative is the size of the peptidoglycan wall. So in gram-positive, the peptidoglycan wall is very thick, versus in gram-negative is thin. So the way I memorize it is that gram-positive has got a really thick peptidoglycan wall. And then the other way that I memorize the other feature of gram-negative is that gram-positive has a thick wall. In return, gram-negative, in order to compensate for that, has developed an extra membrane. So there is an outer membrane that is not present in the gram-positive bacteria. And then the space between the outer membrane and the inner membrane, which is shown here. So the space between outer and inner is called a periplasmic space, which is only present in the gram-negative bacteria, and this space contains the beta-lactamase enzymes, which will break down the beta-lactam antibiotics like the penicillin. And then gram-positive bacteria contains teichoic acid, which is used for attachment to the surfaces. So the way I memorized it is that T, like teichoic acid, has a plus shape, so teichoic acid. In return, gram-negative bacteria contain a lipopolysaccharide in the outer membrane, so the lipopolysaccharides, and the lipid A component of the lipopolysaccharides is toxin and is referred to as endotoxin, which can cause sepsis. So that's the reason that gram-negative bacteria are commonly associated with sepsis, due to the presence of endotoxin in the outer membrane from the lipid A component of the lipopolysaccharide. Next, I would like to go through this table and mention the differences between gram-positive and gram-negative bacteria. So both gram-positive and gram-negative can have capsule and glycocalyx, which will help with the attachment, and the chemical composition of capsule and glycocalyx is the polysaccharide gel, with the exception of the Bacillus anthracis, which has a D-glutamate structure. And then outer membrane, we just discussed, is only present in gram-negative bacteria, and then the lipopolysaccharide component of it, particularly the lipid A, is toxic and is forming the endotoxin. Cell wall is seen in both and is formed by the peptidoglycan. So the way penicillin and cephalosporin work is that they bind to the penicillin binding protein and inhibit the synthesis of the peptidoglycan. Periplasmic space, we discussed, is the space between the outer and inner membrane, which is only present in gram-negative bacteria. And it contains the beta-lactamase, which will break down the beta-lactam antibiotics like the penicillin. And the medications that can be used to inhibit the beta-lactamase include the clavulanic acid, sulbactam, as well as the tazobactam. And then the inner membrane is seen in both gram-positive and gram-negative. So here again, we have the inner membrane in both the gram-positive and the gram-negative, while the outer membrane was only present in the gram-negative. So here we have the inner membrane in both gram-positive and gram-negative, and of course the components of the inner membrane include the phospholipid and the proteins that are embedded inside. And then spores are mostly seen in gram-positive bacteria. It's made of a keratin-like coat, as well as the dipicolinic acid and the peptidoglycan. And spores make the bacteria resistant to drying and chemicals. Next, I would like to go over the organisms that do not stain well with the Gram stain, and these organisms include, for instance, Treponema. So Treponema pallidum is the cause of this syphilis, and these organisms are too thin to be visualized, and thus you will have to use dark field microscopy to visualize them. Mycobacterium, like Mycobacterium tuberculosis, contains too much lipids in their cell wall, as a consequence of which it will interfere with the Gram stain. So for these bacteria, you will have to use the Zeal, Nielsen, Carbol, Fioskin stain to stain the acid-fast organisms. And as a reminder, acid-fast organisms include Mycobacteria, Nocardia, as well as the cysts of the Cryptosporidium parvum, which is a common cause of diarrhea in HIV patients. So cysts of Cryptosporidium parvum, Nocardia, and Mycobacterium stain positive with the acid-fast stain. And then we have the Mycoplasma. So Mycoplasma doesn't stain well with the Gram stain, and the reason for that is that it has no cell wall. So we just discussed that the Gram stain stains the cell wall. So if Mycoplasma has no cell wall, then it won't be stained with the Gram stain. And then we have the obligate intracellular organisms like Rickettsia and Chlamydia, which cannot make their own ATP and will have to live inside the other cells. So these organisms, since they're inside the cells, they won't be stained with Gram stain either. And then finally, Legionella, which is mostly intracellular, doesn't stain well with the Gram stain, and thus you will have to use silver stain for this bacteria. And that concludes our discussion.